channel once again, and welcome to my another video as well. In today's video I'm going to talk about methods of gene cloning, but I'm just going to introduce about the different types of gene cloning methods, and I will try to go in detail for each method in upcoming videos, because if I go into detail today it would be too lengthy and probably you won't digest it. Now, I would like to go with methods of gene cloning, and I have divided them based on the uses of restriction enzyme. First, the method that use the restriction enzyme, and second, that doesn't use the restriction enzyme or use the very minimal amount of restriction enzyme. So, first method is recombinant DNA technology, which use the restriction enzyme. And another type is not using restriction enzymes, the gene cloning methods that doesn't use the restriction enzymes or use restriction enzymes for once. They are Gibson assembly cloning, gateway cloning, topo cloning, and silk. All of them either do not use restriction enzyme or use once. Now I'm going to introduce about the each type or each method of gene cloning here. First is recombinant DNA technology, which is based on using restriction enzyme. Recombinant DNA technology uses the restriction enzyme. As shown in the figure, in the similar way, we proceed the recombinant DNA technology to clone our specific gene. First, what we have to do is we have to do the restriction digest for both donor DNA and the vector DNA. So from the donor DNA, once we do the restriction digest, we do the gene isolation. The gene that we want to clone, we have to isolate using the restriction digest. We cut the donor DNA and we get the fragment, and then we prepare our vector DNA in the similar way. We have to do the restriction digest, and then what we do, we recombine our isolated gene into the vector DNA, and then we ligate them to make a recombination. Ligation always help to stick the gene fragment that we have isolated from donor DNA into the vector DNA. So after ligation and recombination, we have to transform these recombination into the specific host cell. So after we do the transformation after certain hours, based on the like which protocol we followed, we have to select. Do you know the recombination process is not always 100% efficient. All of the recombination may not be successful. So how do we do this selection for successful recombination? Yes, once we grow our transfected cells into the specific media, then we have to observe the colony. We have to do the colony PCR using these specific primers that amplify that isolated gene, mean that gene we have cloned. We have to use this specific primer to amplify this sequence. So what we do, we take the colonies, several colonies, and we have to do the PCR. We have to do the colony PCR. Then we can run this colony PCR result into the agar gel, and we can visualize under the gel dock. In this way, the successful recombination will have this gene that we have cloned or we have recombined. So we will get the band under the expected size. So we can confirm like certain colonies, bacterial colonies have recombination, or if we did not get any band, we can say these colonies doesn't have recombination. So I have taken here an example of bacteria. It could be either E. coli or Agrobacterium. In this way, we have to select the right recombination. After we find the right recombination, we have to regrow them and we have to culture them, and we use for different research purpose. And if you would like to go in detail for recombinant DNA tech, I have made several videos earlier, so you can check with different videos that I have posted earlier. So this was about the recombinant DNA technology. Now I would like to go with another type, the type which doesn't require any restriction enzymes, or if it requires just once. And first example is Gibson assembly cloning. This is known as very easy and convenient method of gene cloning because it doesn't require any restriction digest, and it's just one tube reaction, and it uses dedicated exonucleases as well. But You may be thinking how our gene of interest can be like recombined with the vector without using any restriction enzyme. This is quite interesting thing as well. We have to first create PCR product for our gene of interest that we want to clone, and we have to use a specific primer so that it will create like a homology between our gene of interest and into the linear vector as well. So we amplify our gene of interest with overlapping ends using the specific primers, and we will have the same overlapping ends with the linear vector. So these overlapping ends in between gene of interest and our linear vector will get stick once we transform our or recombine our gene of interest into the linear vector. So we have to take care for using specific primers for gene of interest and linear vector PCR, so that we will have the same overlapping ends in between them. So our recombination will be easy. After creating PCR products, we have to linearize our destination vector. Here, we can linearize our destination vector either using PCR, or we can use restriction enzyme to open up our destination vector. And this much one time is enough for using restriction enzyme here if we're using the restriction enzyme. And then what we have to do is we have to incubate. We have to put our gene of interest that we have done PCR with specific primers and have overhangs. We have to incubate with alpha T5 exonucleases, because it digests back to our vector and PCR product to create overhangs. Then after that we have to polymerize. How we can polymerize is we have to use the Fuchsian polymerase, and we have to follow by ligation. Otherwise, if we do not use the ligase, we might not get the right recombination. The ligase enzyme helps to recombine our insert into the vector, and it helps to stick them together. So in this way we can do the Gibson assembly cloning. It's so easy, and want to experiment, we have to make a PCR product for our gene of interest, and we have to linearize vector. But we have to take care, we have to use the specific primers for both vector and gene of interest to create the same overhangs. So it creates the homology between them. Just only thing we have to consider, then we have to incubate with alpha T5 exonucleases, and we have to do the polymerization and ligation. After the polymerization, then we have to transform our recombination into this specific host cell. It may be either bacteria or any yeast cells or any other virus cells as well. So usually we use the bacteria, and usually we use E. coli and Agrobacterium as a model organism for transformation. So after transformation, we let them grow into the specific media, and all of the transformation and everything based on the specific protocol, which protocol we following. Then after transformation, we probably will get different colonies. Then we have to do the selection. Likewise, I have mentioned in the recombinant DNA technology, we do the transformation to the specific host cell, we let them grow, and we do the colony PCR for selection. I have already mentioned cloning is not 100% efficient at all. Or even if it's 100% efficient, we have to prove in scientific way that we have got the right colonies, right recombination. So what we have to do is, in the same way I have mentioned in the recombinant DNA technology, we have to transform into the specific host cell, let them grow, and we have to select, screen certain colonies, bacterial colonies. If we have transformed into the bacterial cell, then we have to do the colony PCR. We have to use the specific primer to expand or amplify the gene of interest that we have cloned. Then we can see the band in gel with a specific BP size, specific base pair size for the gene. So in this way, or in the similar way we have done in the recombinant DNA technology, we have to do the transformation and selection of our clones. Now, another example is gateway cloning, which also doesn't require any restriction enzyme, and this is known for a very common and convenient and easy method of cloning because it is like highly efficient as well, and it doesn't require any restriction enzyme. But it has two different component. It has like BP reaction and LR reaction. So like unlikely Gibson assembly cloning, for the gateway cloning, first we have to prepare the entry clone, and for the entry clone we should like have our gene of interest with ATTB flank section at the both side. Mean our gene of interest that we want to clone should undergo the PCR using specific primers that makes the ATTB at both sides, and then we should transform or we should recombine our ATTB flanked PCR product that we're going to clone into the donor vector that has ATTP sites and CCDB. So after using the BP clonase, we will get the entry clone. And this entry clone will have our gene of interest that we are going to clone, and it has ATTL site. And once we got the entry clone that has LTTL sites and our gene of interest, we have to transform these gene of interest, or we have to clone our gene of interest into the destination vector which has CCDB and ATTR sites. Then once we have our entry clone having our gene of interest to be cloned, and once we like recombine our entry clone into the destination vector having CCDB and ATTR site using LR clonase, we will get the expression clone. Mean we in this way we make the expression clone. So after that, our expression clone will express the gene of interest, which mean we will get the recombination here. After that, what we do this transformation and then selection. Similarly, likewise, I have mentioned in the recombinant DNA technology and Gibson cloning as well. Once our expression clone will express our gene of interest that we wanted to clone, we have to transform them into the specific host cell, maybe E. coli, based on your which protocol you're following or what you want. Then we have to grow them into the specific media, we have to do the colony PCR, and we have to see the image under the gel electrophoresis system. So in this way we do the gateway cloning, which is so easy and convenient method of cloning. Here is another example, which is topoisomerase cloning, which is also a convenient and easy method of gene cloning. This is also called topo cloning as well. In this cloning, what we have to do is we have to use this specific vector, which is called topoisomerase vector. This is also easy and convenient method of cloning. However, scientists are like using mostly the gateway and Gibson cloning. So in this type of cloning, what we do at the first is we have to create our PCR product, the PCR product for our DNA that like code the specific gene, and we want to clone that gene. So we have to design specific primers to amplify our DNA strand or gene of interest that we want to clone, and we can use any of the PCR product protocol we want to use. So after creating the PCR product for our gene of interest, we have to mix this PCR product with specific vector, which is topoisomerase vector. Then we have to like incubate for 5 minutes at the room temperature. And like some of the topoisomerase vector may have the CCDB gene. So once we like mix our PCR product into the topoisomerase vector, if we have the successful ligation or successful recombination, this gene, CCDB gene, will be disrupted, so that the CCDB gene, once disrupted, then our recombination will be successfully grow into the plate, into the media. So in this way, we can also like verify if we have got the right recombination. So after we mix and incubate, we have to transform this recombination into the specific host cell. Then after transformation, we have to do the selection like I have mentioned earlier in the same way. However, in the topoisomerase cloning, this is convenient and easy. Like if we have the vector like DNA that has CCDV gene, because once we have the vector having CCDV gene, if our recombination successfully ligated, then the CCDV gene will be disrupted and our recombination will happily grow into the media. So we can like directly select that growing colony, and we can like confirm this is our successful recombination. Otherwise, if the gene of interest like did not ligated well, the colony like it won't grow. So we can select in this way as well. And this much about the topo cloning. And here we have another method of cloning which also doesn't require any restriction enzyme, which is called sequence and ligase independent cloning, which is called SLIC. It means it is like independent for using ligase enzyme. It doesn't require to use any ligase enzyme. This is also a unique and like easy convenient method of gene cloning, and it is unique in a sense like it doesn't require ligation. Like for all of the other cloning method, we have to use ligation to ligate our gene of interest into the vector, but it doesn't require it. It's independent for the ligation. So similarly in the silk as well, we have to create the PCR product. And for the PCR product, we have to use specially designed primers so that at the end of our PCR product, it will create the homologous like site as our destination vector. So we have to create a PCR product for our gene of interest that we want to clone, and we have to use a special primer so that it will create the homology between our destination vector and our PCR product. So we have to linearize destination vector either using the PCR, or if you want to use the restriction digit, we can use once here. Now the next step is recombination and polymerization. First, we have to recombine our PCR product, the PCR product for gene of interest, into the linearized vector. Then we have to do the polymerization. For the polymerization, either we can use the Fuchsian polymerase or T4 phase DNA polymerase, because both of them have the 3' to 5' exonuclease proofreading activity, so we can use either of them. After that, we have to add nucleotides. The adding nucleotides or adding DCTP will help to prevent the digestion and will help to promote the polymerization. After that, we have to transform our recombination into the specific host cell, as I have already mentioned in the other types of cloning. And finally, we have to select the right recombination through the colony PCR or any other verification method. So in silk, usually in a lump sum, it depends on specially designed primers that will make the ends of our PCR product homologous to our destination vector. And the exonucleases activity of T4 phase DNA polymerase or Fuchsian polymerase will create the overhangs at the ends of our PCR products and within the homologous sites of our destination vector as well. Thus, it will allow our PCR product, mean gene of interest and vector, to enable to create a final clone piece of DNA. In this way, silk works. And this much for today. Thank you all for keep watching my video till end, and I have tried to explain the different methods of gene cloning that are important in molecular biology. So I will try to like elaborate each type of cloning in my next videos or upcoming videos, and we will see you in next video. Thank you all for keep watching me till end for today's video, and thank you all for your continuous support and motivation. Please do not forget to hit subscribe, like and share button, and we will see you next video. Thank you.